Hello, Ms. Brown here. Today we'll be having a lesson about the cell membrane. And I think something that surprises freshmen when they get to the high school and we learn about the cell membrane, most of their career they've just been drawing this line around a cell and calling it the cell membrane. Well, there is a lot that happens in that line that affects living things. So let's look at our organizer. We'll start on page 1. The definition and location, of course, is the outer edge of cytoplasm of all cells. And keep in mind, you can stop this as we go to give yourself time to get things written in. Okay, and the functions. It encloses the cell like a baggie, like a Ziploc bag. It separates the cell from its exterior environment. And it also controls the substances that enter and leave a cell. It is the gatekeeper. It tells the cell what can come in and what can come out. Now, this is a very special characteristic. So besides special characteristic, I want you to write that it's selectively permeable. So it's kind of picky. It has the ability to decide what comes in and what comes out. And that can change from moment to moment. It might let water in here for a little while and then, nope, don't need any water right now. It might let sodium in, sodium chloride in, just depending on the cell and its needs. It will let certain things in and certain things out, depending on what's best for the cell at the time. And it's composed of 2 parts. And those 2 parts are a phospholipid bilayer, and proteins are embedded inside that bilayer. And I'm going to show you some pictures here to help you get that in your head. So 2 parts, phospholipids and proteins. All right, the phospholipid part, let's talk about it. Phospholipids. They're in 2 layers because bi means 2, right? So hydrophilic means water-loving. So those are the polar heads and they point out. And then the hydrophobic, that means water-fearing. Tails are nonpolar. They're repelled by water and they point in. And small things can actually squeeze through there, but it forms a barrier to most things of the cell. So here you can see I've written page 82. So if you have your biology book, you can look at page 82, but I will show you some pictures also here. So let's look at this one. Here are the heads. They're hydrophilic, right? They like water. And here are the tails. They're hydrophobic. So they're tail to tail. They got their tails pointed in. And notice as you look back here, this gives a 3D structure, how it looks across the top of this. It's almost like a magic carpet or something of these hydrophilic heads. Now, so there's different kinds of proteins that are along in here. So let's look at your organizer. For A, you have channel or transport. That's the name for those. Channel or transport. And they allow substances to cross the cell membrane. It's like a door at your house. You can just walk through. Or a window if you're feeling, I don't know what you'd be feeling to crawl through the window. But this is like a door. The second kind for B, put receptor, and they receive chemical signals. They stick up out of that and they twirl around. They're almost like the satellite dish on your house if you still have satellite, or a TV antenna if you even know what that is anymore. And then C are the marker ones. They identify the cell by type. It's kind of like my house. I live at house 211. The Browns live in house 211 on our street. So it's like a street number. It marks marks the cell what it is. And you need to add D. Those are adhesion proteins, and they help join cell to make tissues to make your organs. Adhesion, add it for D, please, and they join cell to cell. So you can stop this again and write these down. Okay, here's a picture. So here's this phospholipid, the blue. Here's this phospholipid bilayer. We have the hydrophilic heads, the hydrophobic tails pointing in. And now we have these different proteins in here. They're floating around in this. Remember, this is a lipid, and if you remember from our study, a lipid is a fat. So this is a greasy thing here on the outside, repels water. Here we have a transport protein. See this door where things can go in or out? What do you think these are? These are your receptors. They're out there trying to figure out information. So this just gives you an idea of what it looks like. And here it says it's like protein icebergs floating in a sea of grease. So they just drift around and float around through there. And I'm going to show you a few videos so you can kind of see what that means. But it makes it very flexible. It's a flexible membrane. It's not hard or rigid where it can break. Okay, so now I want you to look at number 7 on your organizer. And I want you to label that. You have a little word bank there. So I'm going to give you a few moments, stop the video, and label that, and then we'll go over it. Okay, so using your word bank, for A, you should have embedded protein. For B, you should have phospholipid bilayer. For C, you should have hydrophilic head. And for D, you should have hydrophobic tail. So that's a nice little diagram there to help you remember where all these things are we're talking about. So let's talk about how these things move in and out of this cell membrane. All right, there's 2 ways. Number 8, A, passive. It uses no cell energy. It doesn't require any energy. It's like falling off a log. Okay, and B is active. It requires cells to use energy, like when you climb a hill or a ball rolling up a hill. It's active. It has to have some energy to do that. And this involves what we call a concentration gradient. Concentration gradients. So substances move down the gradient from high to low. That's passive. That's like if I walked in this classroom and I had a big old bottle of stinky smelling perfume, and it would start to move all over the room. It would slowly spread all over the room. That's passive. To change that, you have to have some active or some action to change to make things go up the gradient. If it's already concentrated, to move it up, you're going to have some energy to make that happen. So that's why it's called active. So let's look at our chart here. We're going to talk about small and medium-sized molecules moving into the cell membrane. Okay, so let's talk about diffusion. That's the first one, diffusion. And find in your chart where it says concentration gradient, that's where things go from high to low. So it goes from high to low. Is that passive or active? That's, of course, passive. So in the column there, you put passive for active or passive. And then transport protein required? No. These things can actually slip in between those hydrophilic heads and tails, and they don't need a door or window protein, a transport protein. And no protein is required. And examples of that are oxygen and carbon dioxide. Now let's talk about osmosis. Osmosis is water only. So under examples, water only. Okay. It has to go across a membrane to be called osmosis, and no transport protein is needed. It goes from high to low, and this has to do, we've talked about before, osmotic pressure inside cells, like a plant. If a plant doesn't have enough water, it wilts over because it's lost all its turgor pressure. So if it loses its turgor pressure, we'll have plasmolysis. It actually wilts. If it gets too much turgor pressure, you can get cytolysis, and the cells can actually burst and cause problems for the organism. Okay, the next one is facilitated diffusion. It is again passive, so that means it goes from high to low, but it does need a carrier protein. And an example of this is glucose. So it's a larger molecule, so it has to have some sort of carrier protein. And then we have active transport. And of course, by the very name, we know it's active. It goes from low to high, so it's going up that gradient. It must be assisted by a carrier protein, so yes, a protein is required. And I have 2 examples here. One example is a sodium-potassium pump, so you could do the symbols Na and K, and the proton pump. And this has to do with your body. In your body, one of the things is your heartbeat and keeping the correct amount of these elements in your cells. Now let's talk about large molecules. Let's say it's something too big to slip through there. Okay, you're going to have 2 types. Endocytosis, of course, it's active. So down there in the chart, you put where it says energy required, you put yes. And when it says process required, you put cell membrane engulfs the particles. It just swells around it. Think about Pac-Man. That's kind of what it looks like. It looks like Pac-Man. An example of this is how white blood cells engulf bacteria in your body. They just eat it up and digest it. So it brings things in. And there's 2 kinds. There is, here on your page, there is phagocytosis. Now, freshmen, I know you're going to want to run saying phagocytosis, but it's phagocytosis. So beside that, I want you to put cell eating. And then there's penocytosis, and beside that, I want you to put cell drinking. So there's two kinds of endocytosis. And now we have exocytosis. This is how large molecules get out of the cell. Again, it's active, so where it says energy required, yes. And then under process required, vesicle fuses with cell membrane. So it actually goes to the side and fuses, and then it opens up, and that substance is released. Okay, so it goes out. Then over here under examples, excretion is wastes. And then secretion is usables. Now let's look at the bottom here where it says, refer to the diagram. This one-celled amoeba is eating its food by, and what would that be? Which one was cell eating? Look at your chart. Of course, that's phagocytosis. And it's similar to the way human white blood cells ingest germs. And somewhere there beside that little drawing, I want you to write, it's like the video game Pac-Man to help you remember. Okay. And that's the end of part 1.